Hello everyone, welcome back to my channel, the Biolab Scholar. So in my previous video, I discussed about the importance of restriction enzymes, and I also discussed how you can choose the order of restriction enzymes that you are going to add in your forward and reverse primer. So in this video, I'm going to discuss how you can actually find the restriction enzymes that you can use for your gene of interest to clone in a desired entry vector. So let's get started. So I'll start sharing my screen as usual so that you can see how I am doing it. Okay, so again, as an example, I'm taking the same ACT1 gene that I used in my video where I showed how to design the primers and how to name the primers so that it is easier for you guys to learn how you add the enzyme sites with the same primers. So once you have your gene of interest amplified using the PCR, you actually know the precise size of your insert, which is very important. And now you will see why I was emphasizing so much that you need to know the actual size and sequence of your gene of interest. So once you know the precise size of your insert, we are going to take that sequence and check which enzymes do not cut in your gene of interest, and that also corresponds to the multiple cloning site in your entry vector. So we let's suppose that we have used the forward primer from the ATG, and we have taken the reverse primer from the stop codon. So I am going to copy this whole portion, and now I have to check the enzymes that do not cut in my sequence. So I'll go on Google and then I'll search for NEB cutter. So NEB cloner is a great site for any new beginner. They have all the protocols and everything for a beginner. So you can also consult those guides. And for today's video, I'm going to use one of the online tools from NEB cloner, which is NEB cutter. So I'm going to open NEB cutter, and then once it is open, I just have to paste my sequence in the box, and then I'll just submit my sequence. So now here you can see a graphical view of your gene of interest and all the enzymes that cut in between your sequence. So for now, the default option is being selected, which is one cutter. So all the enzymes that you see here cut one time in between your gene. And then if you select two cutter, then you will have the options, all the enzymes that cut two times in your gene of interest. But what we want is, we want to choose the enzyme that do not cut in your gene of interest and also present in your multiple cloning site of your entry vector. So let's suppose like ZO1 and EcoR1, we are looking for that do not cut in your gene of interest. So I will go on zero cutters. You can see here that the zero cutters is being selected. And now I'll just scroll down and look for the enzymes that are present on my MCS and also do not cut in the gene of interest. So I'll just scroll down and I'll try to look for EcoR1. Okay, this is great. I find that EcoR1 do not cut in my gene of interest. And then the other enzyme was ZO1. So I'll just scroll down. This is all alphabetical, so you can just scroll down and go. So, okay, great. ZO1 is also here. This also doesn't cut in your gene of interest. So assuming that the orientation of the enzymes in your multiple cloning site is ZO1 is first and then EcoR1. So we are going to add ZO1 site in the forward primer and EcoR1 enzyme in the reverse primer. So let's I'll show you how we add the sites. So the sequence for ZO1 is CTCGAG. I'll go back in my primers, and because I started using this primer that starts with the ATG, I'm just going to add the enzyme site. So CTCGAG. Let's highlight so that we can track later which one is the enzyme site. Maybe I'll just highlight with the highlighter. Okay, now I know that I have added the enzyme site in my forward primer, and I also have to add the enzyme site on my on my reverse primer. And you have to remember that whenever you design your primer, we always write down sequence from 5' to 3', because 3' is the one that extends in the PCR. So whatever enzyme sites we are adding, we add it on the 5' end so that they are the overhangs, and in the next in the second cycle of your PCR, the overhangs are added in your sequence. So I'll go and check what is the sequence for EcoR1, which is GAATTC, I think, but I'll just cross check. Oh yeah, so it's GAATTC. So I will go and then just add GAATTC. So I'll also highlight this. I think, yeah. So now we have both the primers with the desired enzyme sites flanking a sequence. Here, once we have added the enzyme sites, our name of the primer also changes. Until now, like how we have named Arabidopsis thaliana ACT1 1 forward. This primer doesn't give you any information about the enzymes that you are going to add in your sequence. So once you are using your primer for cloning and you have an enzyme site over it, you have to mention the enzyme that you are going to add. So the one will be the primer name. So the primer name also changes as you add the enzyme site in your primer. So similarly, for the reverse primer, we are going to write R1 reverse. So just by looking at the primer, now we know that we have amplified this long fragment, like 104 base pairs long fragment, using ZO1 and EcoR1 as the flanking enzyme sites. So now that we have added the enzyme sites on the primers, the work is not yet done, because some of the enzymes also need some additional base pairs for their proper activity. So we also have to look for if our enzymes that we have used for that we are using for cloning also need such nucleotides or not. For that thing, you again visit the NEB cloner, and then you have to choose the option. Like if you go at the home of New England Biolabs, you will choose the option tools and resources, usage guidelines, and in the cleavage close to the ends of DNA fragment. In this option, you are going to get a PDF about the oligonucleotides that needs to be added at the ends of your enzyme sites. So I have like already downloaded, this is a PDF downloadable file. I have already downloaded this file and I'll just show you in the PDF format. So here this table is showing you all the enzymes plus all the nucleotides needed by the enzyme according to the reaction that you are going to use. If you are doing your digestion for 2 hours, this is the column that you have to refer. If you are digesting for over 12 to 16 hours, this is the column you have to add. And then depending upon the efficacy, you have like different options if you have to add only single or dinucleotide or trinucleotide. So it all depends. So we will go and reach the EcoR1 enzyme. So here you can see that for all the digestions, the efficacy is more than 90%. So in this case, I always prefer to use the trinucleotides, because the more nucleotides will be there, it is the efficacy is better. So if this is the option where there is no difference, I will go for trinucleotides. So, and then we know that we have to add the sequence on the 5'. So then we will add CCG and not CGG on the 5 prime. We have to add on the 5 prime side of the enzyme side. So we'll add CCG. So I'll go back and then I'll also add CCG. So now this primer is complete. This is your full primer that you need to order in order to amplify your gene for cloning. So let's also check for ZO1. So I'll go back to the PDF and just scroll down until I reach ZO1. And then, okay. So here you can see that there is like a huge difference between the efficacies in like 2 hours and 20 hours reaction. So I'll go for the highest one. If I'm doing a reaction for like overnight, I do generally overnight digestion reactions, but you can also do like 1 hour, 2 hour. So in the overnight reaction, this 75% is the highest, and which is reached by the trinucleotides. So I will add again, this is same as EcoR1, but then of course we have to check every time because the sequence also differs in like different enzymes. This isn't coincidence. There's ZO1 and EcoR1 have the same extensions. So I'll add CCG to my forward primer as well. So yes, now we have both the primers ready for order. And then one more thing I wanted to mention is that why do we need to know the actual sequence? So as I gave you example for designing another forward primer from the 5' UTR, and if you do not keep the information about the sequence that you included in your insert, you might miss out including this sequence when you check your enzyme sites in your insert. And then there are chances that this sequence might have the enzyme that you are going to add in your primer. So if you do not include this site to check if there are enzyme sites in your gene of interest, you are going to add them in your primer, and then later when you will be doing cloning, you are not going to get the correct sequence. Like when you will do the sequencing, you are not going to get the results, and then you might wonder like what went wrong. So this is like very important that you keep a track of actual nucleotides. Even like leaving a single nucleotide can make a difference. So it is very important that you keep a track of every single nucleotide you are including in your gene of interest. So yes, that's all for designing the primers and including the enzyme sites in your primer. And now these primers are ready to amplify your gene of interest and do the further cloning. So I hope you find the video helpful, and please do share, subscribe, and comment if you find it, if you like the video. Till then, take care, stay happy, stay healthy. Bye-bye.